Hey guys, hope you're having a really good week so far. Um, Today we are going to be talking about the senses of hearing and balance. Again, this is our last lecture video for this semester. This is your the last uh, new information in lecture. We will be finished with chapter 15, which is our last chapter for this semester, so we're almost done. I'm sure you guys are excited to hear that. Again, tomorrow you will have a quiz over chapter 15, and you will also be working on our last lab exercise for the semester, and that will be lab exercise 23, which is over hearing and balance. And then remember on uh, Thursday, we will be having our last lab quiz, which will be over lab exercises 23 and 24. I'm sorry, 22 and 23. And then on Friday, we will have our last lab exam, which will also be over exercises, lab exercises 22 and 23. That is hearing and vision. And ideally, I would have also had lab exercises 17 and 18 because those are over the brain and spinal cord on this lab exam as well. However, like I mentioned previously, I forgot and included them on our lab exam 3. So lab exam 3 was really long. It was over 100 points if you noticed on Infinite Campus. But our lab exam 4 will be a lot shorter because it's only going to be over these two. I was also, if we had been in school, we were going to be dissecting an eyeball and a brain. But since we're not in school, we don't get to dissect those. And so this lab exam is only going to be over lab exercises 22 and 23. And so therefore, again, it will be shorter and smaller, probably not even worth 100 points. Again, so the last two lab exams, 3 and 4, will probably average together to give you about 100 points for each one. Our last one, our lab exam 3 was like 120 something points, I think. So we'll have obviously less than that on our lab exam 4. It'll be probably 100 points, but together they'll average out to be about 100 points piece total. And then, of course, so after this Friday, we are done with everything except our last lecture exam. We will have our last lecture exam on, well, it'll be next week. Okay, it'll probably be, I'm trying to think, probably go ahead and do that on Monday. And then the rest of the week, you'll have some time to go back and review the material, and then I will assign you your final exam around the end of next week. We were supposed to do it. We're supposed to be done by May the 11th, which will also be the next Monday. So I'm thinking probably next Friday is when I would do the semester exam, or I'm sorry, this semester exam, your final. The only thing is, I need to. I may even ask you guys a question on one of your quizzes this week or something. When do you want to do your final? Because I know you may want to go ahead and get it over with. So, but yet, if you want to do it sooner or later, I want to give you that time to look over things and study. So, it's up to you. I will probably let you. I'll probably ask you a question on one of the quizzes toward the end of this week. Would you rather do your final like Tuesday or Wednesday, or would you rather put it off until the end of the week? And then whatever I get as far as majority rules, which there's only 3 or 4 of us, so therefore, whichever one wins, I will let you know that for sure. So by the end of the week, I will be letting you know for sure when your final will be, because I know, like I said, some of you would rather get it over with. Some of you might want to have more time to look over things before we do it. So I'll be getting your input on that and let you know something by the end of this week. So let's go ahead and look at our senses of hearing and balance. We're going to see that our ear not only allows us to hear, which we all know, but we may or may not have thought about. Our ear also helps us to keep our sense of balance. You can you've heard of people who have an inner ear infection many times. Not only do they have pain, but also it makes them really sick because their head spins and they can't walk a straight line. They can't keep their balance. And the reason for this is because our inner ear is responsible for our sense of balance. It's responsible for giving us information about the position of our head in space. And so if there is an infection in that inner ear, then that messes with our sense of balance, and this is what causes the spinning, the head spinning, the dizzy feeling, and not being able to stand up and walk straight and things like that. Okay, so first we want to look at just the anatomy of our ear, and then we'll look more at how it functions to help us both hear and keep our sense of balance. So our ear itself is divided into three parts: the external ear, middle ear, and inner ear. The external and middle ear are involved only in hearing, while our inner ear, as I just described earlier, is involved not only in our hearing but also in our sense of balance or our equilibrium. The parts of our external ear, first we have the flap of our ear, which is on the outside of our head. That's called the auricle or the pinna. If you remember, it is composed of elastic cartilage covered with skin, and it does have a function. Its main job is to collect the sound waves and funnel them into the external auditory canal. So that pinna or auricle actually acts as kind of like a funnel, collecting all the sound waves that it can and funneling them into that external auditory canal. The external auditory canal is lined with hairs and with ceruminous glands. Remember, ceruminous glands produce a secretion called cerumen, which is earwax. So the main function of this external auditory canal, not only does it pass the sound waves collected by the pinna down into the middle ear, but it also, those hairs and earwax help to, they're like a sticky trap. They help to trap foreign objects and prevent them from reaching and building up on down there in the middle ear on that eardrum. Now our eardrum is actually the border between our external and middle ear. So it kind of forms kind of a barrier, not really a barrier, but it's the boundary between the external ear and the middle ear. Our tympanic membrane is another name for our eardrum. Our eardrum is the layman's term. The technical word is tympanic membrane. It is composed of 2 layers of epithelium that have a little bit of connective tissue between them to hold them together. And when those sound waves get to the end of that external auditory canal, you have the eardrum there, the tympanic membrane, and those sound waves will cause that tympanic membrane to vibrate. So again, what we're looking at here, guys, is we're looking at vibrations. We're looking at mechanical sound waves, and we're going to see that the sensory receptors that turn these sound waves into into what we hear are those mechanoreceptors, because it's these vibrations that will be stimulating these mechanoreceptors in our inner ear to generate action potentials that are sent along our vestibular nerve and our cochlear nerve, which we'll talk about those more in a few minutes as well. So here we can see our entire ear. Again, our external ear is all the way here to this tympanic membrane, which is our eardrum. Then we have our middle ear right here, which is basically going to include these auditory ossicles. We'll talk about those in a second. And then the inner ear here is this spiral organ, as well as these semicircular canals. The spiral organ is called the cochlea. So again, here is our funnel, our auricle or pinna, that collects those sound waves and funnels them into this external auditory canal. And then here at the end of this external auditory canal, we have our tympanic membrane or eardrum that will vibrate when those sound waves hit it. Okay, moving on to our middle ear. Again, our middle ear, of course, is separated from our inner ear by our tympanic membrane. It's separated from our external ear by the tympanic membrane, and then it is separated from the inner ear by the oval and round windows, which we will be talking about shortly. Inside that middle ear, you have 2 passages for air. You have your auditory tube or eustachian tube that opens into your pharynx, which remember is right there at the back of your mouth. It's kind of the top of your throat. And this is just important for equalizing pressure. Like when you're going up a mountain or coming down a mountain or a very high hill or maybe an airplane or helicopter or something, you will feel this pressure. And then many times, I know for me, I can just swallow. Sometimes they say chew gum, but I can just swallow and then that pressure will go away. And that's because when I swallow, that kind of equals out that air pressure. It kind of passes some of the pressure that's built up into that eustachian tube, and equalizes out that air pressure between the outside and the middle ear, and that relieves that pressure. So again, your ears are connected to your throat. And and you probably already knew that. Your ear, nose, and throat doctor. Uh, your ear, nose, and throat doctor, that you have an ear, nose, and throat doctor because all of those things are connected together. So our ear is connected to our throat by this eustachian tube or auditory tube. Also, if a child has an ear infection, a middle ear infection, it's called otitis media. Many times they may have a scratchy throat or a sore throat. And drainage as well, because the excess fluid from that infection actually drains down the eustachian tube into the throat and drains down the throat. The other passages for air is a passage that connects to the mastoid air cells in the mastoid process. So again, the mastoid process is going to be there, part of that mandible and temporal bone. Probably, I'm not sure if it's the mandible or the temporal bone. It might be the temporal bone. In any case, yeah, I think that's what it is. It's the temporal bone. But anyway, you have air cells inside that bone that also help to have to deal with pressure there in the middle ear as well. The main parts of our middle ear are the auditory ossicles. We call them, the layman's terms are hammer, anvil, and stirrup, but the technical terms are the malleus, incus, and stapes. I remember malleus because I think of a mallet, which is like a hammer. I remember stapes, stirrup. They both start with ST. The stapes is the stirrup, and then of course then the other one is the incus, which is the anvil. And what these are going to do, guys, they are connected to the eardrum, and they are also connected to the inner ear by that one of those windows. I don't know if it's the round or oval window. It's the oval window. Is the connection between the middle and the inner ear. So anyway, so these auditory ossicles are connected both to the tympanic membrane and to the oval window, which is the connection to the inner ear. So vibrations are transmitted from the tympanic membrane. Okay, remember these are vibrations from sound waves. They are transmitted from the tympanic membrane, which is the external ear, to the oval window, which is the inner ear, by these auditory ossicles. These auditory ossicles form a physical connection between the external ear and the inner ear so that they can transfer the vibration. So the tympanic membrane will vibrate. This in turn vibrates the auditory ossicles, and then this in turn vibrates the inner ear. And again, that's what we need. We need the vibration of the inner ear, because remember, it's those there's in that inner ear there are mechanoreceptors that will be stimulated to generate action potentials when vibrations when they bend. These actual sensory receptors are called hair cells, and they bend. And when they bend, they generate action potentials. Well, what bends them? Well, it's the vibrations from the external ear that were transmitted to the inner ear by these auditory ossicles in the middle ear. There are also a couple of skeletal muscles in the middle ear. These are called the tensor tympani, which is attached to the malleus, and the stapedus, which is attached to the stapes. And these are two skeletal muscles that help to just kind of dampen, like I think it's called sound attenuation. They help to dampen like really loud sounds as it goes through that middle ear. And then of course, as I just mentioned, you have the oval window that is the connection between the middle ear and the inner ear. So again, we have the auditory ossicles that are connected between to the tympanic membrane on one side, and then on the other side, they're connected to this oval window, which is the connection to the inner ear. Specifically, the stapes, okay, the stirrup, the stapes, the foot plate of the stapes rests in the oval window, and it is held there by something called the annular ligament. So the foot plate of the stapes rests in the oval window, and specifically the vibrations of that stapes will be transmitted. So again, the vibrations get transmitted from the tympanic membrane to the, I think it's the incus, then the malleus, and then the stapes. So they're kind of in a row, and we'll see that right here. Here you can see your incus, which is your anvil. Here you can see your malleus. Here's kind of the head, and here's the handle of the malleus, the hammer. And then here's the stapes, and here's the foot plate of the stapes resting in the oval window right here. You can also see the tympanic membrane, the eardrums, or the eardrum here. This is going to be your external auditory canal. So the vibrations will come through here. They'll hit the tympanic membrane, and they'll vibrate. It'll cause vibrations, and then it'll vibrate these auditory ossicles. And then this foot plate of the stapes rests in the oval window, and that is the connection to the inner ear, which we will be looking at next. You can also see here the tensor tympani attached to the hammer. You can see the stapedus attached to the stapes. There's that external auditory canal, also called the auditory tube. You can see some ligaments. Again, these are ligaments holding bone to bone. So you can see a ligament here. You can see one here. You can see one here. You can see this is a tendon, again, holding the muscle to the bone. And here's another tendon holding this muscle to this bone. So again, those bones are held in place by ligaments and tendons connected to muscles. Okay, so now let's look at the inner ear. In the inner ear, we have two labyrinths. We have the bony labyrinth and the membranous labyrinth. The bony labyrinth just consists of tunnels and chambers in the temporal bone, and it is lined by endosteum. So the bony labyrinth is actually connective tissue called endosteum that lines the tunnels and chambers in the temporal bone. We also, part of this bony labyrinth includes the cochlea, which is the organ of hearing, the vestibule, which is an organ of balance, and the semicircular canals, which are also organs of balance. Again, these are all connected together. The cochlea is actually spiral, and then you have the vestibule that is kind of in between the cochlea and the semicircular canals. And then the semicircular canals, there are 3 of those, and we'll see that in just a minute. Okay, the other labyrinth is the membranous labyrinth, and these are membranous tunnels and chambers that are suspended in the bony labyrinth. Okay, so the bony labyrinth, again, is endosteum lining tunnels and chambers in the temporal bone. Okay, inside that endosteum, inside that bony labyrinth, we have membranous labyrinth. So these are like 2 layers. This one is going to be a little bit more external. This one's a little bit more internal. So it is just membranous tunnels and chambers that are suspended within the bony labyrinth. And suspended within those, we have the 2 fluids. The endolymph is the most internal fluid, so it's inside this membranous labyrinth. And then the perilymph is going to be another fluid that is in the spaces between the membranous labyrinth and the bony labyrinth. Okay, so here is our all of this together. Again, the bony labyrinth is more external. Again, this is endosteum that's lining the tunnels and spaces in that temporal bone. And then inside that, we have this membranous labyrinth. So the perilymph is going to be out here, and then the endolymph will be inside here. Okay, and we'll see some more pictures. Also, you can see here's our spiral organ. That's the cochlea that is used for hearing. Okay, this is the vestibule. And then here are the 3 semicircular canals that are, and we'll see they're almost at right angles to one another. Okay, here we can also see the liquids or the fluids inside these 2 labyrinths. Again, inside of the membranous labyrinth, which is going to be here in pink. Here in pink is the membranous labyrinth. So inside of that, between the membranous labyrinth and the bony labyrinth, which is here kind of in this brownish gray color, we have the perilymph, and then we have more perilymph in here, and then I'm looking for the endolymph. Okay, the endolymph is here inside the membranous labyrinth. So again, between the bony and the membranous, the bony is the brown, the pink is the membranous, we have this blue perilymph. And then inside the membranous, so inside the pink, we have this lighter pink, that is the endolymph. And again, this is just a cross section through one of those semicircular canals. So all of this contains this, same material. This is the whole organ. It's responsible for both hearing and balance. Again, this spiral part is the cochlea. This is the vestibule, and then these 3 tubes here are the semicircular canals. We can also see the round window and the oval window. Again, remember the foot plate of the stapes rests in this oval window. And that is what when it vibrates, it transmits vibrations from the middle ear to the internal ear here. This is the internal ear. Okay, 3 distinct regions of the cochlea, which probably is referring to this whole thing is called the cochlea. So the 3 distinct regions are the scala vestibuli, which is from the oval window to the helicotrema, which is the apex of the cochlea, the scala tympani, which is from the helicotrema to the round window, and then the cochlear duct, also called the scala media, which is formed by the membranous labyrinth and is filled with the endolymph. So again, Um, Back in here, we have the scala media. The scala tympani is going to be between the round window and the helicotrema. So that's going to be like right around in here. And then the scala vestibuli is from the oval window to the helicotrema. So again, that's going to be right around in here. Okay, the scala vestibuli and the scala tympani are filled with perilymph. We've already talked about that. That is going to be this material in here, the blue. And the oval window communicates with the vestibule, which communicates with the scala vestibuli of the cochlea. So again, the oval window communicates with the vestibule. So here's our oval window. Here's our vestibule. This gray round structure is the vestibule. The oval window is right there, kind of within that vestibule. And then that vestibule communicates with the scala vestibuli of the cochlea. Okay, the wall of the scala vestibuli is called the vestibular membrane. The wall of the scala tympani is called the basilar membrane. And what we see is that the width of this basilar membrane is going to be thinner near the oval window and then thicker near the helicotrema. And the reason for this thinner basilar membrane responds to higher frequency vibrations, and then the thicker basilar membrane responds more to low frequency vibrations. So the differences in thickness of this basilar membrane help our ear to be sensitive to both high frequency and low frequency vibrations. So again, here we're going to see the round window and the oval window. This is the cochlea. Here are semicircular canals, and here is our vestibule. The helicotrema is here kind of in the center of the vestibule. And then we can see here we have this basilar membrane here, and we can see how that it is going to be, looks like, thinner here. You can see how thin it is here. Again, that is more toward the oval window. And then toward the helicotrema, which is more back this direction, you can see how it is thicker. Again, allowing us to be sensitive to both higher frequency sounds and lower frequency sounds. Okay, the spiral organ, which is the organ of Corti, contains the sensory cells that are responsible for hearing. So again, in here, this cochlea, this is the spiral organ or organ of Corti, is where the cells that are used for hearing are located. And these cells are called hair cells. These sensory cells that are located inside that spiral organ are called hair cells. And they have little projections that are microvilli at their apical end. So this is their free end, not the end that's attached to the membrane underneath, but the free end. They have hair-like projections, but they're not cilia, okay? They're called stereocilia, but they're not cilia. They are microvilli. Remember, cilia are more like hairs, whereas microvilli are actually extensions of the cell membrane that don't necessarily move around, okay? These can bend, but they don't really move around. But so it's kind of misleading. It makes you sound like, oh, these are cilia, these are little hairs, but again, they're not actually little hairs that are embedded in the cell membrane. They're actually microvilli, which are extensions of the cell membrane. So a little bit of distinction there. And they are called stereocilia. The tips of these inner hair cells are embedded in something called the tectorial membrane. And then you actually have two rows of these hair cells, inner and outer. The inner hair cells are embedded in the tectorial membrane. The outer hair cells are involved in regulating the tension of the basilar membrane. And we'll look at more about that in a minute. However, so you have these two layers of hair cells, inner and outer. You have something called a tip link that connects the stereocilia, each stereocilium to the one next to it. So you have adjacent stereocilia. One is longer than the other, and the tip link is a little gating spring, is what we're going to call it, that attaches one stereocilium to the next one that's longer than it. And it also attaches to a potassium ion channel that opens mechanically. So basically what you're going to see when vibrations bend these stereocilia, it pulls on that potassium ion channel, and that opens that potassium ion channel, and then that causes calcium ion channels to open, and this ends up resulting in depolarization of the hair cells. It ends up resulting in depolarization, which results in an action potential in these hair cells. So here we can see the stereocilia. We can also see how you have shorter ones and then longer ones, and you have those little tip links, or those, what do we call them? These gating springs that when these bend, it causes one to pull on the next one, which ends up opening the potassium ion channels, which then opens the calcium ion channels. Here you can see a diagram of this. Again, you have two layers of hair cells. The hair cells are arranged in rows. You have inner hair cells that that actually are responsible for hearing, and then you have outer hair cells that regulate the tension on the basilar membrane. A hair bundle is all of the stereocilia of one hair cell. So like this would be a hair bundle, okay? So because this is all the stereocilia associated with one hair cell. And then we have this structure here called the tip link, which is a gating spring that attaches the tip of one stereocilium in the bundle to the next longer stereocilium. And then again, as these are bent, they open potassium ion channels, which are mechanically gated ion channels. So this is how vibrations end up in an action potential. Because when these bend, they mechanically open these potassium ion channels. They physically pull them open and allow potassium, and then when potassium comes in, this opens up the calcium ion channels, and then when calcium comes in, this causes depolarization of the hair cell. So again, you can see it here. This tip link is connected to this potassium ion channel. So when the stereocilium bends, it pulls on that tip link. So let's say it's probably going to be bending in this direction, and it pulls that little spring-like molecule, and when it pulls it, it mechanically, physically pulls open this potassium ion gate, allowing potassium to go into the cell. And then when potassium goes into the cell, this triggers the opening of calcium ion channels. And when calcium ion channels open, calcium comes into the cell, and this is what results in the depolarization. So similar to the way that sodium ions coming into the cell open up the skeletal muscle, cause depolarization of the skeletal muscle. Well, when calcium ion channels open up and calcium comes into the cell, it's going to cause depolarization of the hair cell. The thing here is we have to open up potassium ion channels first. When potassium comes into the cell, then this is going to trigger the opening of the calcium ion channels. Okay, inside of our inner ear, we have a basilar region of hair cells that is covered by the synaptic terminals of sensory neurons. Specifically, these are going to be sensory neurons from the cochlear nerve. So you're going to have synaptic terminals, those little feet at the end of these sensory neurons that are going to cover the basilar regions of the hair cells. So, and then the cell bodies of these sensory neurons are grouped into cochlear ganglion. Okay, so remember, a ganglion is a group of cell bodies. So cochlear ganglion contains cell bodies from these sensory neurons. So one end of the sensory neuron has synaptic terminals that are associated with the hair cells. So the action potential in the hair cell will be transferred to the action will then stimulate an action potential in the sensory neuron. And then the cell bodies of these sensory neurons are grouped into cochlear ganglia. And then the fibers of these sensory neurons form the cochlear nerve. The cochlear nerve is connected to the cochlea, okay, and that's for hearing. We're also going to have a vestibular nerve that's connected to the vestibule, and the semicircular canals are all there connected together. You have a vestibular nerve that's connected to the vestibule that is more for balance, and so both that vestibular nerve and that cochlear nerve come together to form the vestibulocochlear nerve, which we also call the auditory nerve. Again, it's not just for hearing, but it's for your sense of balance as well. So here we can actually see the synaptic terminals of the cochlear nerve associated with the basilar region of this hair cell. Here is our hair bundle with those stereocilia. Again, shorter ones, longer ones, connected together by those tip links. And so that's kind of what we just talked about. And then, of course, in our spiral organ, our organ of Corti, which is that spiral that we saw earlier. We saw that that is where these hair cells are. And again, here is the hair bundle. And then here is your tectorial membrane as well. Remember, those stereocilia are embedded in that tectorial membrane, which is kind of deep to the vestibular membrane. Okay, so let's talk about how hearing actually works. Your external ear, that pinna, is going to collect the sound waves and funnel them into that external auditory canal toward the eardrum, the tympanic membrane. When the sound waves track the tympanic membrane, which now we're in the middle ear, it's going to cause vibrations. When the tympanic membrane vibrates, it vibrates those auditory ossicles, the malleus, incus, and stapes, and then the foot plate of that stapes transfers those vibrations to that oval window. Something else we want to look at is the fact that you guys went through all the way up through the middle ear. We are vibrating sound. Or I'm sorry, we're vibrating air. The sound waves are vibrating through air. Okay, when we get to that middle ear, inside that cochlea there is liquid, there's fluid. And it takes more force to vibrate fluid than it does air. So we have a size difference between the tympanic membrane and the oval window. The tympanic membrane is much larger than the oval window. Also, the foot plate of the stapes is much smaller than the tympanic membrane. So the fact that we have a very large tympanic membrane, it's about 20 times the area of the oval window, and because the foot plate of the stapes is much smaller than the tympanic membrane. So the tympanic membrane is very large compared to the foot plate of the stapes and the oval window. So basically what that does is that concentrates the vibrations. But instead of using concentrate, which I use concentrate because I thought that might make you understand it better, but it amplifies the vibrations. It makes them stronger. We've got small vibrations in this huge tympanic membrane, but those small vibrations in that really big structure become many vibrations, many stronger vibrations in that little bitty foot plate of that stapes in that oval window. So the size of the tympanic membrane compared to the size of the foot plate of the stapes in the oval window helps to concentrate or amplify those vibrations, make them stronger, about 20 times stronger as they pass from the tympanic membrane across the auditory ossicles to the oval window. Because now we are vibrating in that cochlea, we have to vibrate fluid. So we need those vibrations to be much more frequent and much stronger to vibrate that fluid. Again, as we said earlier, the tensor tympani, which is connected to the hammer or the malleus, and the stapedius muscle, which is connected or to the stapes, reflexively dampen really loud sounds. So they attenuate the sounds. This is called the sound attenuation reflex. Basically, they help to contract if the sound is too loud to kind of help protect the inner ear structures from very loud noises. So with those little muscles help to prevent the very loud, really strong vibrations from very loud sounds from damaging those auditory ossicles, as well as the cochlea and things like that inside your ear. Okay, so once those vibrations, those really strong vibrations, get to the inner ear, they are going to vibrate that perilymph. Okay, remember that was that blue inside of the scala vestibuli. And this causes the vestibular membrane to vibrate, which causes vibrations in the endolymph. So again, basically we have the perilymph and the endolymph right there very close to one another. So the vibration of the perilymph, which is more external, is going to cause vibration of the endolymph, which is more internal. So basically these are both fluids inside that cochlea. They're going to be vibrating together like they're one fluid. And this is going to displace the basilar membrane, which is going to then bend those stereocilia, causing those tip links to pull open those potassium ion channels, letting potassium in, which then triggers the opening of the calcium ion channels, letting calcium in, which then causes a depolarization of those hair cells. So again, if we go back and we look at this picture here, Okay, so we've kind of done this step by step. Vibrations go in here. They are funneled in here. They vibrate the tympanic membrane. Again, we can see the tympanic membrane is really big compared to this foot plate of this stape. So when this vibrates, it vibrates the hammer, it vibrates the incus, and then it vibrates the stapes. Again, the stapes is a lot smaller than the tympanic membrane. We can also see here that this oval window is a lot smaller than that tympanic membrane. And so the small oval window, the small foot plate of the stapes really helps to concentrate or amplify those vibrations. So when the vibrations get here, okay, they're going to vibrate first the perilymph and then the endolymph. And then once we start vibrating that endolymph, we're going to vibrate this basilar membrane. Remember, it's thinner here toward the cochlear end, and it's thicker toward the oval window end. Again, allowing us to pick up, to detect both high frequency and low frequency sounds. So we're going to vibrate that basilar membrane, and then remember that that is going to bend those stereocilia, and when this stereocilia bends this direction, it's going to pull on that tip link, which is going to open those potassium ion channels, let the potassium in, and then when potassium comes in, that triggers the opening of calcium ion channels, letting calcium in and causing depolarization. Okay. Again, so here this is showing it to us. Again, here's our vibrating of our tympanic membrane, vibrating our hammer, our anvil, and here's our hammer, here's our anvil, here's our stirrup. Here's the foot plate of that stapes, that stirrup in that oval window. These are a lot smaller than that big tympanic membrane, so we get a lot stronger vibrations to vibrate this perilymph and endolymph inside this cochlea. And then these vibrations are going to travel along that basilar membrane. Again, different places, allowing us to pick up different sounds, different high frequency, low frequency, different pitch sounds as those hair cells, as the stereocilia on those hair cells are bent. Again, different frequency sounds can cause maximum vibrations of different regions of the basilar membrane. So again, here's that spiral organ of Corti, that cochlea. And again, we can see here how different frequencies, these hertz, okay, 20,000 hertz. Again, that is there at one end of that cochlea. That's the very thin basilar membrane. And then as we go more up toward the apex of that cochlea, the apex of that spiral organ, the basilar membrane is thicker, so we're picking up more low-pitched sounds, the lower frequency sounds. So vibrations of stereocilia on the hair cells of this membrane at different areas of this cochlea are allowing us to pick up different pitches, different frequencies of sounds. Again, higher pitches near the base, lower pitches here near the apex. Again, the apex is kind of the center of that spiral organ. Again, we can see here at the base around in here, we have the very thin basilar membrane. At the apex up in here at the end of this spiral, we have a lot thicker basilar membrane, allowing us to pick up those lower pitch sounds. So again, what happens once we get the sound to the nerve, okay, to the vestibulocochlear nerve? So again, when it goes from the hair cells to the cochlear nerve, then that combines with the vestibular nerve, giving us the vestibulocochlear nerve. Again, those action potentials then in the vestibulocochlear nerve will generate action potentials in the cochlear ganglion. And then these will travel to the cochlear nucleus of the medulla oblongata, then to the superior olivary nucleus. So remember, we have olives in the medulla oblongata that help with the modulation of sound. We also have, from the olives of the medulla oblongata, it will go to the inferior colliculi of the midbrain. If you remember, we talked about, and I brought up the brain PowerPoint here, the inferior colliculi are involved in hearing. These are part of the midbrain. And then they'll go to the medial geniculate nucleus of the thalamus. We also talked about that with the brain. If we go to the thalamus, there we have our medial geniculate nucleus involved. It is the part of the thalamus that receives the auditory information. Remember that all senses except the sense of smell go through the thalamus before they go to the correct area, the correct area of the cerebral cortex or the cerebrum. And so it is the medial geniculate nucleus of the thalamus where our auditory information will go. And then from there, it will go to the auditory cortex of the temporal lobe. Again, remember, our auditory center is in the temporal lobe of the cerebrum. Another place that some of these signals from what we're hearing go is, remember, to that the inferior colliculi. If you remember, we talked about these yesterday as well. The inferior colliculi are part of the midbrain. And so not only does it go to the inferior colliculi, but it also goes from the inferior colliculi to the superior colliculi, which is responsible for those reflexes, such as when we turn our head toward a loud sound. So you hear a really loud sound and you turn your head and your eyes toward that. Well, that is because of the superior colliculi. They are involved in not only visual reflexes, turning your eyes toward something that's really bright or a loud sound, but also those auditory reflexes where you turn your head toward that loud sound as well. That, remember, those are reflexes that happen. And so these impulses from our vestibulocochlear nerve, after leaving the medulla oblongata, they go to the midbrain, not only to the inferior colliculi, but also to the superior colliculi. And that's how we do those reflexes of turning our head and our eyes toward a loud sound, for example. And then they go there before they go to the thalamus, before they're filtered by the thalamus, and then go to the temporal lobe, which is the auditory center of the cerebrum. And this is just a visual with little numbers showing you everything we just talked about, from the vestibulocochlear nerve up to the superior olivary nucleus, which is the olive of the medulla oblongata, and then on up to the superior and inferior colliculi of the midbrain, and then from there to the medial geniculate nucleus of the thalamus, and then from there going on to the auditory cortex in the temporal lobe. And also here somewhere you can see where it went to the inferior colliculus can actually go back to the ear as well, and I'm not sure about all that. Okay, so don't worry about this part really, just this main part, from the vestibulocochlear nerve to the olives of the medulla oblongata to the inferior and superior colliculus of the colliculi of the midbrain to the medial geniculate nucleus of the thalamus, and then to the auditory cortex. That's the main one. I'm not worried too much about that number 5. Okay, so now let's talk about our sense of balance real quick. You have two labyrinths, the static labyrinth and the dynamic labyrinth. The static labyrinth contains, it's basically two parts of the vestibule, the utricle and the saccula. The utricle and the saccula of the vestibule make up the static labyrinth, and they evaluate the position of your head relative to gravity. So remember, gravity is always pulling down. So they are evaluating the position of your head relative to gravity, and they also help you to detect linear acceleration and deceleration. So, you know, when you are moving forward, moving backward, like when you're in a car. And then your dynamic labyrinth is made up of your semicircular canals, and these evaluate the movement of your head in three-dimensional space. So, you know, like when you bend over and you stand back up, or you're rolling down a hill or something like that. The dynamic labyrinth, those semicircular canals help you with that. So, let's first look at the static labyrinth, which are going to be the utricle and saccula of the vestibule. The utricle has the utricular macula that is oriented parallel to the base of your skull. And then the saccula has the saccular macula, kind of a tongue twister there, that is oriented perpendicular to the base of your skull. So again, the utricle macula is oriented parallel to the base of your skull. The saccular macula is oriented perpendicular to the base of your skull. And again, remember, these are in that vestibule of that inner organ, of that inner ear. These macula, both of these, the utricular macula and the saccular macula, contain specialized epithelium that contains supporting columnar cells with hair cells with numerous stereocilia. So guys, these are a lot like the hair cells that were in the cochlea. Okay, they're going to be very similar. They have these stereocilia, which are microvilli. Again, they are not actually cilia. They are those extensions of the cell membrane called microvilli. And it also has one cilium called a kinocilium. And remember, in our cochlea, we have those stereocilia, those hair cells, stereocilia, embedded in that, what was the name of it right here? That tectorial membrane. Okay. Well, in our vestibule, the stereocilia and the kinocilium are embedded in something called the otolithic membrane, which is a gelatin. It's a gelatinous mass. And in that gelatinous mass, that otolithic membrane, you also have these little crystals composed of protein and calcium carbonate called otoliths. So they're almost like little stones. But again, they're not rocks. They're not stones. They're little crystals, real tiny crystals that contain protein and calcium carbonate. So in this otolithic membrane of gelatin, we have stereocilia and the kinocilium embedded in that gelatin, and there's also otoliths, or little like stones or crystals in that gelatin. So this otolithic membrane, this gelatin, moves in response to gravity. Okay, so when you bend over, when you roll around, when you move forward and backward, this gelatin moves. This gelatin inside the vestibule, and we'll look at that vestibule here in a minute, it moves. And when that gelatin moves, then those hair cells are moved. They are bent. Just like the vibrations from the middle ear to the inner ear vibrate that perilymph and that endolymph, which is a fluid. Okay? Well, those are vibrations from sound that bend those stereocilia of those hair cells. Well, here, when you move your head, it moves that gelatin, that otolithic membrane, which then bends the stereocilia of those hair cells embedded in that membrane. And again, these are going to function much the same way as the stereocilia of the hair cells in the cochlea. There are tip links connected to potassium ion channels. When those stereocilia bend, it pulls that tip link, which opens up that potassium ion channel, which is then going to open calcium ion channels to let calcium in and cause depolarization and action potentials. When the hair cells bend toward the kinocilium, this causes depolarization. When the hair cells bend away from the kinocilium, it causes hyperpolarization. So again, a combination of depolarization and hyperpolarization depending upon which way you move your head and which way those stereocilia bend, whether they bend toward the kinocilium or away from the kinocilium. If the head is tipped, then otoliths move in response to gravity and stimulate certain hair cells, and again, cause action potentials, cause depolarization and action potential of those hair cells, which sends information to the brain about the position of your head in space. These hair cells synapse directly with neurons in the vestibulocochlear nerve. So just like the hair cells in the cochlea synapse directly with neurons of the cochlear nerve, well, the hair cells with the stereocilia in the vestibule synapse directly with neurons from the vestibulocochlear, or from the vestibular nerve, which is going to combine with that cochlear nerve to form the vestibulocochlear nerve. So again, here we see, remember, this is our vestibule here. Okay, all of this area here is our vestibule. And you have your utricle is this portion of the vestibule. So all of this is vestibule. And your utricle is this portion of the vestibule. Your sacula is this portion. Okay, this blue jelly looking stuff, that's that otolithic membrane. And the little crystals you see inside, those are the otoliths. Okay, here we have our hair cells with those hair bundles, with those stereocilia. And here we have the synapse of the hair cell with the synaptic terminals of that vestibular nerve. Here we have a close-up view. Here's a hair cell. Here is the stereocilia. Here is that kinocilium. Again, it's on that on the end of the taller stereocilia. And then here we have the synaptic terminals of the neurons of those vestibular nerves synapsing directly with those hair cells. As these otoliths in this jelly-like otolithic membrane move in response to gravity, the intensity of hair cell stimulation stimulation, it changes. So again, the more you move your head, the more that otolithic membrane moves, which then bends those hair cells, which bends the stereocilia of the hair cells embedded. The stereocilia are embedded in the otolithic membrane. The pattern of stimulation and action potentials that are generated as a result of the bending of the hair cells as this otolithic membrane moves as you move your head is going to be translated by your brain and tell you about where your head is in position to the rest of your body and in space. And a lot of this you don't even, it's subconscious. You just know without thinking about it. You know, okay, I bent my head to the side, I bent forward, you know, my head is moving forward, my head is moving backward, kind of thing. So here we can see that when you're looking forward, your head is straight up. Your otolithic membrane is in such a way that the hair cells are, the stereocilia of the hair cells are being bent in such a way that you know, I mean subconsciously you know your head is up facing forward. And then when you bend over, you bend your head over, then that changes the orientation of your vestibule. It causes that otolithic membrane to move. And it's going to bend the stereocilia embedded in that. Here they are. You see the little hair-like things embedded to be bent in a certain direction. And that pattern of bending and stimulation of those stereocilia, that pattern of information is sent to the brain, and then your brain interprets that as, okay, now my head is bent over and I'm looking at the ground. Okay, so that's the static labyrinth. The static labyrinth has the otolithic membrane and the otoliths and the stereocilia on the hair cells that move in response to you moving your head. Okay, now we're going to look at the dynamic labyrinth, which is the semicircular canals. As you can see here, we have 3 of them, 3 semicircular canals. They are nearly at right angles to one another, and they are all filled with endolymph. Okay, so again, fluid. Here's our vestibule. We are moving that otolithic membrane, that jelly inside of that. Well, here we have fluid again. So we have jelly in the vestibule, which is the static labyrinth. We have fluid in the dynamic labyrinth. Again, we have a transverse, coronal, and sagittal planes of these semicircular canals. So like this one is in the transverse plane. If you know the transverse plane, it's more horizontal. This one is in the sagittal plane. So remember, that's like cutting straight, dividing into left and right half. So it's going to be more vertical. And then the coronal is the one like the coronal plane or the frontal plane. So it's like flat, cutting you into front and back. So that's going to be this one right here. The base of each one of these semicircular canals is expanded into what we call an ampulla. So 1 ampulla, 2 ampulla, 3 ampulla, 4 ampulla. These are ampulla. They're just the expanded portions at the base of the semicircular canals. And inside that, you have a sensory epithelium called the crista ampullaris. The crista ampullaris is very similar to the macula, the epithelium in the macula. You know, that utricular macula and the saccular macula of the vestibule. So again, in here in the utricle and the saccular macula. Inside this crista ampullaris, you have, so here's your crista. We're doing a close-up view of this little structure right here. So this is the ampulla around it here. Inside, we have the crista ampullaris, which is the epithelium, and in that epithelium, you have these crista hair cells. Okay, so again, we have more hair cells, and they are embedded in this cupula. The cupula is this blue structure here. It acts as a float, and it is displaced by fluid movements in the semicircular canal. So we have the hair cells, again, with their stereocilia embedded in the cupula. The cupula is like a little float that moves in response to the movement of the fluid in the semicircular canals, the movement of that endolymph in the semicircular canals. So when that fluid moves, so there's fluid around here, like this clear would be fluid. When that fluid in the semicircular canals, that endolymph moves, this moves this cupula. And when that cupula moves, it moves the hair cells. It bends the stereocilia of the hair cells again, opening up potassium ion channels, which lets potassium come in, opens up calcium ion channels, which lets calcium comes in, which causes depolarization and generates action potentials. displacement or movement of this cupula is most intense when the rate of head movement changes. So, like when you bend over real fast or you bend back up really fast. And this allows this entire system to detect changes in the rate of movement of the head rather than just movement alone. So not only are you detecting movement of the head, but also the rate of movement of the head, such as really fast versus really slow. So again, when you slowly bend over and stand back up versus bending over very quickly and standing back up. So again, for balance, hair cells, the stereocilia of the hair cells in the macula, that's the saccular macula and the utricular macula, as well as the stereocilia of the hair cells of the crista ampullaris, when embedded in that cupula. When the stereocilia of those hair cells are bent, then that generates action potentials that are going to be transmitted to the vestibular ganglion, then join the cochlear ganglion to form the vestibular nerve, then goes to the vestibular nucleus, again in the medulla oblongata. Will go to this information will go to the spinal cord, to the cerebellum, to the cerebral cortex, and to the nerves controlling reflexive movements of the eye muscles. And those are going to be those lateral rectus, superior rectus, inferior rectus, medial rectus, as well as the superior and inferior oblique muscles of the eye, which are controlled by the oculomotor, trochlear, and abducens nerves. So information from this vestibular nerve and vestibulocochlear nerve will actually also go to the oculomotor, trochlear, and abducens, and this is what allows you to maintain visual fixation on an object when your head is in motion. So even though you're moving like in a car, you can focus on something because some of this information about the movement of your head from this vestibulocochlear nerve is going to the oculomotor, trochlear, and abducens nerves. Okay, and then this is just a visual showing you the same thing about the pathway that the sense of balance takes to get to your brain. Again, from the vestibular ganglion here's the vestibular ganglion along the vestibular nerve to the vestibular nucleus in your medulla oblongata here. Then that's going to go to the cerebellum, which influences postural muscles. Then it's going to go to motor nuclei here that help to control your eye muscles, helping you to visually fix on an object when your head is moving. Then from there, it's going to go to the posterior ventral nucleus of the thalamus. And then from there, it's going to go to your cerebral cortex, a couple of different places, the vestibular cortex in the temporal lobe, and there's also part of that in the parietal lobe as well. Okay, and that will be it for our senses of hearing and balance. Please let me know if you have any questions. Again, tomorrow you will have a quiz over Chapter 15, and you'll be doing your lab exercise 23 over hearing and balance. Then on Thursday, you're going to have a quiz over lab exercises 22 and 23. It'll be pretty short. And then on Friday, you will have your last lab exam 5. Again, it will be on lab exercises 22 and 23. And then I'm also going to be asking you to let me know what day next week you would rather have your final, whether you'd rather have to do it, pretty soon after your lecture exam 5 over chapters 12 through 15, which we will have on Monday, or if you would rather do it later in the week, like on Thursday or Friday, giving you some time to look over things. So, and then that will be the last 2 things we'll have to do after this week. So please let me know if you have any questions. Hope you have a great day and a great rest of your week, and I'll be talking to you later. Bye-bye.